Okay, I think we're live. Let's just see. Good morning, everyone out there. I think that we are live. I'm just going to wait and get some confirmation on my end that we are streaming. Excellent. We are live. Well, good morning, everyone. Welcome to our Facebook Live. I'm Morgan Itis. I'm the Manager of Education at Fighting Blindness Canada. And I'm very excited today to be here with Dr. Brian Ballios. And we are actually together in person in New Orleans at the Association of Research in Vision and Ophthalmology Conference, which is very exciting. And we're here to sort of celebrate National DNA Day, which is tomorrow, April 25th. And this year is actually a big one. It's a 20th anniversary of the completion of the Human Genome Project and the 70th anniversary of the discovery of the DNA double helix structure. So it's a pretty cool time, and also just being here at the conference, being surrounded by all of this exciting research, and it's a really great day to have this Facebook Live. So I know many of our regular followers know Dr. Ballios very well, but Dr. Ballios is a fellowship-trained clinician scientist who focuses on medical retinal disease and the subspecialty in inherited retinal and genetic disease. He is an assistant professor at the Department of Ophthalmology and Vision Scientists at University of Toronto, and he holds staff appointments as clinician scientist at the University Health Network in the Donald K. Johnson Eye Institute, and is a staff physician at the Kensington Vision Research Center. He's also a scientist at the Crumble Research Institute, and he has a laboratory where he's investigating the mechanisms of retinal disease and the development of new stem cell-based therapies. You're a busy man, Brian.
Very busy. Thanks for having me.
Well, why don't we dive right in? And again, if there's questions today, please feel free to put them in the chat, and we'll try to answer them. But we're going to start by just going back to the very basics. So what is DNA? What is a gene?
DNA, deoxyribonucleic acid, is really the building blocks of life on Earth. Multicellular organisms have DNA as their genetic code, and it's the blueprint for life. DNA tells our body how to make different proteins and enzymes that are important for how our cells function every day. And DNA is important for how all the cells in our body work, not just the cells in the eye or the retina, but important for how the cells in our brain work and how the cells in our gut work and how the cells in our skin work. So DNA is an incredibly complex blueprint that has carefully evolved over millions and millions and millions of years of evolution to make us who we are today. And there's an incredible amount of intrinsic variability in our DNA and genetic code. And that intrinsic variability accounts for all of the different differences that we see amongst individuals in terms of our appearance and the way we think and the way we act. So the other important feature of DNA, of course, is that it's heritable. I mean, we get our DNA from our parents, and DNA is passed down through the generations, and it has been for millions of years. It's a characteristic feature of life. So DNA is really, really important in understanding DNA, and that milestone of mapping the human genome was a huge landmark in human evolution.
And so what specifically is a gene when we're thinking about DNA?
So a DNA is the raw code, right? And it's a sequence of letter code that is billions of codons long. And that genetic DNA is organized into genes. And genes you could think of as little books. Or one way I try to describe to my patients is a gene is like a novel. It's like a book. And that book has a title, and that book has a name. And that book tells our body it's the story of how to make a protein, a very specific protein inside the body that has a function. And just like books are divided up into chapters, genes can be divided up into chapters. And genes are simply a way of how that DNA is organized. And so we start to think about conditions that affect human health and genes. We start to think about how misspellings in the DNA within specific genes, perhaps in chapter 1 or chapter 13 or chapter 9, how a specific misspelling in a word might change the whole way that the body reads that book and makes a protein.
So specifically when someone has like an inherited retinal disease, what is happening there with the genes?
So we think that there's over 20,000 genes that are important for how the human body works. But we know that only in a subset of those genes is the genetic code really important for how the retina is structured and how the retinas functioned. And we think that there's about 320 to 330 different genes that are important for how the retina works. And in an inherited retinal disease, there's a crucial misspelling in part of that genetic code within one of those genes, within one of those books. And we call them inherited retinal diseases because often, by far and away, the most common way of getting that misspelling in your DNA is inheriting it from a previous family member. And for many of these misspellings, they've been present for many, many years throughout human evolution, through many generations of a family, perhaps. And they're passed down through the generations in different ways. Sometimes they're hidden in our genetic code, and we need to get one misspelling in a copy of the gene from our mother and from our father in order to have the disorder. And sometimes one misspelling is enough, and we can get one from a parent who had a condition, who got theirs in turn from a grandparent who had a condition, and on.
So it's very complicated, obviously. And so we're thinking about treatments and things, and now these gene therapies are starting to get more attention. So how does a gene therapy treatment work?
Well, so the revolution in understanding inherited retinal diseases over the last 20 to 30 years has been really identifying the different genes that cause the heritability. Back in the 50s and 60s, and even in the 70s and 1970s, clinician scientists and physicians, they understood that these disorders were heritable through the families because they had family members. They could see these disorders were passed down. They didn't know the underpinning and why those conditions were having an effect on the retina. And so through the 90s and into the 2000s, we've learned so much more about how these genes actually cause disease. And as we learned about it, we found that the vast majority of retinal diseases are caused by a single misspelling in a single gene. And as opposed to some other genetic disorders where it's a confluence of multiple misspellings of different genes, most genetic retinal disorders are single gene disorders. We call them monogenic. That just means single gene disorder. And as we learn more and more about the way those genes contribute and the misspellings in those genes contribute to disease, we started to realize that maybe if we had a way of putting back a properly spelled copy into a light-sensitive cell, perhaps in the retina, that has a misspelling, then maybe we could restore the function of that protein and then restore the way that the retina works. And that's really the basis for the different gene therapy approaches. Now, gene therapy is a broad field. There's many tools that people are using to try to correct those misspellings or put back a properly spelled copy of the gene. But in principle, the idea is get a properly spelled copy of the gene back into the cell. And there's been some great laboratory evidence to suggest that will improve the function and health of the cells. And now, excitingly, there's a lot of evidence in humans in clinical trials to suggest that indeed that approach can be very helpful.
Yeah. So is that why I would say, like, right now we hear a lot about gene therapy? It's everywhere. Is that why it's getting so much attention right now? Because this development is there's a lot of clinical trials to show that there's some evidence that this is working.
Yes. So, in fact, one of the very important reasons it's getting a lot of attention right now is that there has been a successful gene therapy. The first FDA-approved gene therapy for use in humans was a product called Veretagene Naparvovec, and the trade name for that is called Luxturna. And that was a gene therapy that was developed, and it took many years, decades development, all the way from laboratory studies through to testing in humans to show that for a very specific misspelling in a very specific gene that's important for the retina, a gene called RPE65, it's an enzyme that's important for how the retina works. A misspelling in that gene that a patient inherits from their mom and a copy of misspelling they inherit from their dad means that they don't make functional enzyme inside their retina. And that gene therapy is able to put back a properly spelled copy of that gene and thereby restore the function of the retina. And that's helping out many patients with this disorder. And it's an incredible proof of concept for how gene therapy could work to improve and restore and preserve the health of vision for these patients. And so now, because of the exciting success of that gene therapy, um, investigators, scientists, industry collaborators, everyone's become very interested in what other genes could we potentially repair or treat with gene therapies. And so that's led to a proliferation of clinical trials in this space. And that's one reason why I think it's getting a lot of attention.
Absolutely, and very exciting. Luxturna now approved by Health Canada and is now available, I think, in many provinces, publicly funded, which is great. So you'd mentioned that Luxturna is the only. Are there any other approved therapies for gene therapies for eye disease, or this is the only one right now?
For inherited retinal disease, this is the only one right now, and there are several. There are dozens of clinical trials going on for other gene therapy approaches for other genetic retinal disorders, and they're at various stages of development. And gene therapy trials have to be done in a very careful way through phases, including understanding whether they're even safe to use in humans. I mean, we think they are if they go to a clinical trial because there's lots of laboratory evidence. You never know for sure until you try it in person. And then through various stages, assessing the efficacy and how much potential benefit it has to the retina. But I think the important thing to remember about clinical trials is they really are a form of controlled experiment in humans. They're not therapies that we know will work. They're therapies that we hope will work. And we are encouraged by the success of similar therapies, the success of specifically Luxturna as a therapeutic. You never know until you do the study how it's going to have an effect in that unique disorder. And just like all of us are different from each other, these genetic retinal disorders are very different from each other in terms of how they progress and the symptoms patients have, what they look like clinically when patients come to visit us. So that can change a lot of things for different clinical trials.
Because correct me if I'm wrong, but even within like retinitis pigmentosa, for instance, there are tons of genes that can cause the diagnosis of retinitis pigmentosa, right? It's not just one gene mutation that causes it.
So retinitis pigmentosa is one form of genetic inherited retinal dystrophy or retinal disorder. And we think that there's over 70 genes that are important for how retinitis pigmentosa might develop. In other words, 70 genes in which we've been able to identify misspellings that can lead to retinitis pigmentosa.
And thinking about Luxturna and some of the clinical trials that is the treatment just preserving existing vision, or is it able to restore some vision as well?
No, I mean, obviously the hope is to preserve the vision that patients have at a minimum. What we've seen in Luxturna is, in fact, patients can get a boost in their vision, in aspects of their vision that I think people who designed the original clinical trials never really anticipated having that much benefit in. Right. Patients who have retinitis pigmentosa or who have retinal disease caused by misspellings in the RP65 gene, many of them experience significant and profound difficulty with their night vision or vision in low ambient illumination. And what we found is that after treatment with RPE65 gene therapy, patients have a tremendous boost. In fact, they can go out and do things in those sorts of lighting conditions, ambient light, that they never could have done before. And that was surprising to give them back that function and to see such tremendous improvements. And in some clinical trials that are going now, been able to see improvements that are also similarly unexpected. And that's, I think, one of the things that really motivates us in this space is discovery, not just finding what we expect to find, but hopefully uncovering and lifting the rock on new benefits that we never expected.
Yeah, that's something I found really interesting at the conference too this week, like hearing about those unexpected benefits. It's not just about like what the best corrected visual acuity is, it's how it affects the life of the patient and their ability to function. So I think that's really cool. So are all of the gene therapies that are being looked at, are they all for very specific gene mutations, or are there some that are looking more broadly?
There are different genetically oriented therapies that are trying to get away from treating one single gene, one single misspelling. And I think one very promising approach, or one approach that many investigators are moving forward with clinical trials, is the approach of optogenetics. And that uses a toolkit in gene therapy to try to regenerate light sensitivity to the retina. Usually or we think at this point, again, discovery, we think at this point it'll be for patients who have lost the majority of their light sensitive cells. So their retina is not as light sensitive as it used to be. And those light sensitive cells, we call them photoreceptors, those light sensitive cells, when they go away over time in the disorders, the body can't regenerate them and make new ones. And in gene therapy, we're trying to, in traditional gene therapy, we're trying to improve the function of those light sensitive cells. But when they go away, we don't have the cell to improve the function.
Right.
So using the toolkit of optogenetics, we use a gene therapy to introduce a light sensitive molecule, a piece of DNA that encodes for a light sensitive molecule, into the helper cells of the retina, the cells that normally communicate with the light sensitive cells, and the cells that are left behind when the light sensitive cells go away. And the idea behind optogenetics is to make those cells light sensitive, thereby providing some vision to patients who've lost those other cells.
It's really interesting. It's fascinating work. Now that's still preclinical work, right? Or though there's been some clinical work too.
There are some clinical trials going in patients who, starting obviously with patients who've lost the vast majority of their vision, the vast majority of their light sensitive cells. And so those clinical trials are ongoing in various countries.
It's very exciting. Yeah. So I will also throw it out there if anyone has any questions. We'd be happy to take a few questions before we go. But lastly, what are you excited about? Like, what, again, being at the conference, what gene therapies are sort of on the horizon? Any clinical trials that you're particularly interested in following?
I'm always interested in all the different clinical trials that are going on for different reasons. And that's really part of the reason I'm interested in the different clinical trials. What they're doing is all of them are slightly different in the way that they're assessing what we call endpoints, or measures of success in terms of measuring vision. And in clinical trials in inherited retinal disease or in genetic retinal disease, this understanding of how the retina works and how we can assess its function has really led to a revolution in how we conduct clinical trials in ophthalmology, and what we use as the benchmark for success. And so there's some very exciting trials in X-linked retinitis pigmentosa. There's some very exciting trials in retinitis pigmentosa caused by misspellings in the gene GUCY2D that affects patients from a very young age and has many features that are similar to the RPE65 retinal dystrophy. And so it's exciting to see the progress in those clinical trials. And of course, it's exciting to see the preliminary data that's presented that is getting people on board with moving to more advanced trials, hopefully to approvals, and we'll have to see what happens. But one of the things I'm excited about any of the trials is understanding how they're assessing their outcomes. We're changing the way we think about how we measure improvement in retinal visual function. And to be very concrete about it, for decades, the gold standard for improvement in visual function was improvement in best corrected visual acuity. In other words, what we measure on the eye chart when a patient comes to clinic, how many letters they read on the eye chart. And we know that for many, many inherited retinal disorders, that simply doesn't reflect the entire burden of challenges that patients have. And there are many disorders for which patients have very well-preserved reading vision, or even very strong reading vision, and yet they have significant dysfunction and challenges because of the way the rest of the retina has trouble. And we need to be able to assess changes in those aspects of vision. And we need to make those aspects of vision and the changes in those aspects of vision important to regulatory bodies like Health Canada in the States here where we are today, to the FDA, Food and Drug Administration. We need to show them that maybe there's a new gold standard that's not just how vision changes on an eye chart. Patients who receive Luxturna gene therapy, patients who are enrolled in clinical studies who are seeing some benefit from gene therapy, are not necessarily reporting that the big benefit is in how well they read, but they're reporting benefit in other things, like how well they navigate and their side vision, their night vision, or maybe less difficulty with their day vision, glare. I mean, these are real symptoms that patients have and they talk to me about in the clinic. And these are things we need to get a handle on in the trials. And we're getting a better handle on it, and seeing those presentations here at ARVO, at other meetings, give us pause and give us a lot of food for thought on how to design better trials to get these drugs approved.
Yeah, I think it's really interesting because it shows that this is really pioneering, right? We haven't done it before. It hasn't been done. So we're learning, even if there's, things that are not necessarily considered a success, we're learning a lot from those trials.
We like to think we know a lot in medicine. There's a lot of things we don't know. And we are learning all the time, not just from the sort of laboratory-based studies, things that I do in my lab, but we learn from our patients too. And they have a lot to teach us. And I think, really we have to put behind us the days when patients were really seen as just subjects in the clinical trial. And we really are listening much more to what patients are telling us, and patient-reported outcomes, PROs, as they're called for short, but essentially listening to the patient and hearing their reflection on the benefits they've had in gene therapy. These outcome measures, codifying them and creating them in a way that we can actually measure effect, is becoming much more important in how we design clinical trials. And we're really pushing regulatory bodies to recognize that data as important data for efficacy.
It's interesting that the advocacy component has to really come in as well at this point.
Side by side.
Yeah, exactly.
Advocacy and the conduction of the clinical trials.
Yeah. Excellent. Well, let's see. I don't think we have any questions, right? Oh, actually, we do have a couple questions.
Okay.
This might be a little specific, but let's see.
It's okay.
So
happen.
Mal is asking if if there's any progress for inherited retinal gene ICQB1.
ICQB1. I know of a lot of laboratory-based studies for ICQB1, but I'm not aware of a clinical study that's going on right now.
Yeah,
I know of a lot of investigators and even some who are presenting data here at ARVO about how they're trying to develop gene therapies for ICQB1, but it hasn't yet come to the point where we're enrolling patients in a clinical study. And that jump from getting the evidence for how efficacious a therapy could be to actually going and trying it in humans takes often the interest of many different parties and some investment of money to move it to that stage. So
yeah.
I'm very hopeful for ICQB1 today.
Is ICQB1, is it a RP or is it?
It is a gene which can cause, we're getting away from more and more from describing it in traditional terms like retinitis pigmentosa,
Yeah.
these old clinical terms that we had in the 50s, 60s, and 70s when we didn't understand the genetics. Right. We found that often patients who have misspellings in these genes have very unique concerns with their vision that don't necessarily fit into the neat bins. So more and more I prefer to think of it as, for example, ICQB1 associated retinal dystrophy. Right. And really just think about those patients as a group of patients.
And I guess that really highlights too the importance of genetic testing, like making sure you have the testing. And even if you're not getting a result now, like knowing that there's new genes being discovered all the time. Right?
In my opinion, it's medical standard of care for working up a genetic retinal dystrophy. You should try to find the DNA cause of why. And it helps us at 3 levels. As I tell my patients, it helps us, number one, to perfectly define the disorder in terms of exactly what gene is causing it. And it takes out the doubt that sometimes patients have and providers have when there's been multiple referrals as to what could be going on in the retina. It helps us really define it concretely. The second reason is, of course, it helps us understand what the risk might be to other family members of having the disorder. And then, of course, the third point, as we've been talking about today, is the impact on potentially new therapeutics.
Yeah, interesting. So we have another question here from Nancy asking if Luxturna works for wet AMD.
Now, Luxturna is a product very specifically developed for misspellings in the RPE65 gene. And so it's not expected to work in disorders that don't have misspellings in that gene. Mm-hmm. And the RPE65 associated retinal disorder has very unique features which are distinct from wet age-related macular degeneration. So no, Luxturna is not for treatment of wet AMD.
So I seem to recall seeing some things this week about gene therapy trials for dry AMD. Mm-hmm. Are there other trials for wet AMD that you know of?
Yeah. So there's clinical trials going on to try to develop gene therapies for wet AMD. And many of those gene therapies are less about trying to put back a misspelled copy of a gene. Many of them are about trying to introduce a gene that might encode for a beneficial protein. Mm. For example, a gene that might encode for the molecule that we inject for patients with wet AMD in the clinic. Molecules like ranibizumab which is known as Lucentis, or aflibercept, which is known as Eylea. These products that are approved for use in humans.
So the anti-VEGF.
The anti-VEGF, anti-vascular endothelial growth factor, anti-VEGF molecules. There's some evidence to suggest that we could, if we introduce those molecules into the retina in a gene therapy approach and have the retina make its own anti-VEGF molecule, maybe we could avoid the burden of having to bring patients back repeatedly for injections for these medications. I mean, those injections have been incredibly successful in many cases at holding back the and staving off the progression of wet macular degeneration. But they're not perfect,
right?
And they require a shot in the clinic, and they require patients to come back for repeated shots. So,
yeah, for sure. Well, very exciting stuff. Okay, I think that's all of our questions for now. Thank you so much for taking the time to talk with us today.
Happy International DNA Day.
Happy International DNA Day. And have a great rest of your day to everyone out there. Thanks so much for joining us.
Bye.
Bye.